heart as a muscle, the heart as an organ, we talk about the conductive system, we talk about the contractile system. And then we started to talk about different volumes and determinants like cardiac output and stroke volume and so on. And we're going to finish the cardio part, the heart part today, by talking about how we regulate this whole big machine that we learned is can be so complex and so important. And the basic idea, just to remind you, is that CO, which is cardiac output, basically tells you how much in shape you are, how efficient your heart works. Pumping the blood to the lungs and to the tissues. Of course, it's more challenging to pump the blood into the tissues, and we will talk about it today, about the vascular system. We'll just start with the vascular system. We'll continue on Thursday. And we learned that our CO is pretty amazing. It's about 5+ liters per minute. But it can go much, much higher if you need. If you, for example, go and exercise, or if you just even go up the stairs, you need a bit more blood. So today we'll talk about some positive and negative regulations. And the basic idea that we always need to remember, and remember, this is a lot about logic. Don't try to memorize as much as you want to actually understand the logic behind it, is that the stroke volume, okay, who wants to remind me what the stroke volume is? What does it mean? Not the equation, the formula, we can see that here. But how can we define stroke volume? Anyone? Cool. Out of blood in a minute, not a minute, in one beat, in one cycle, exactly. The rate is where we put the heart rate. That's where we get cardiac output. So the stroke volume is the EDV, end-diastolic volume. That is the volume in the ventricles, and we will use the left ventricle here. Yeah, but you can go to the right as well. And the end of diastole, right? So just before systole. This is the full max volume of blood inside the left ventricle. Taking away the ESV, what's left? That means how much was ejected, was pumped out of the ventricle into the aorta, large arteries, and tissues. Okay. Cardiac output takes the stroke volume, times it with heart rate, how many beats we have per minute, and that's our cardiac output. Any questions about this before we go and talk about regulation? Just to make sure we're all on the same page. I know it's been like 5 days and it sounds like forever. Okay. So if we want, for example, to increase, and it can be decreased, right? But we'll just focus on increase as an example, stroke volume. Cardiac output. We can increase stroke volume, we can increase heart rate, we can increase both. If we focus first, and we will focus first on the stroke volume, we can either increase the EDV or decrease the ESV, the amount is left, and it's and/or, right? They're mutually exclusive. We can do both. That's exactly what we're going to do. There are going to be 3 factors. The ones labeled in blue are the ones that positively regulate, that increase the cardiac output, and the one in red is going to decrease preload, contractility, and afterload. I'm just going to give you here the sentences. I'm not going to even stay for 2 seconds for this slide. You have it, because we're going to address each and every one of them. Okay? So let's talk about the preload first. Preload is the amount of tension of the ventricular muscle before it contracts. Please note the before. I put it in highlight and bold and blue and everything. So here's the muscle, right? And this is a section, so we can see the chambers, the atria and the ventricles. And this is the muscle. I'm showing it right now with my laser point. And this is the main thing that is going to contract, right? The ventricular muscles, in order to push the blood out of the ventricles. Now this is the resting heart with a solid line that you can see here. And here's the ventricle, what you can think about it as a pool filled with water from the atria, right? Now there is a limited amount of water in the pool or blood in the ventricle that you can put in, but you can also make the pool a bit bigger. How do you do that? You can do it by the fact that the muscle can expand, the wall of the muscle can expand, and these are the dashed lines that you can see. So these are potential sizes of the muscle wall. Now let's think it just for a second, without going into the details. If we are increasing the muscle as a whole, before we talk about contraction, just talking about the size, do we increase the EDV or decrease the EDV? We increase, right? We just made the pool larger, basically. But the issue is that here we're not just talking about the amount, we're talking about the tension developed by the muscle. Do you remember when we talked about, back when we talked about the sliding filaments theory, we had this really challenging question on what happens if you stretch the muscles before they contract, and we said that too much, no tension, overlap great, at some point too much overlap. We talked about that ratio between the length. The same goes here, the same, it's a muscle. Myosin and actin filaments. So if you stretch it to a limit, of course, not too much, you increase the amount, the number of myosin heads that can potentially bind to actin filaments, and you'll create a greater tension. And that's called the preload. How much you stretch, so it will provide you with tension. It's called the Frank-Starling's law of the heart. Frank Starling was not a scientist. Frank was a scientist. Starling was a scientist. Both of them are Frank Starling together. Frank-Starling's law. The more stretched the resting fibers, the more forceful the contraction. If you pull, then you get a more forceful contraction. Yes? Sorry, again?
Are there folds in the ventricle and stuff that allow?
Yes, you have enough flexibility, you have elasticity to pull them. And what's pulling is the wall itself. Okay? So, what did I want to ask about this before we go on how we do that? Yeah, okay, that's basically the idea. So, if we just agree that we can make a stronger tension and we want to provide a strong contraction, right? How do we actually do that? Are we going to put our hands inside our mouth, going all the way to the heart and just pulling all of these? Not so efficient, right? Do we have muscles, you can imagine, or maybe some Cody or something that will pull? That can be a great idea. But that's not the way it works. The way it works is that we're going to push, not pull, but push the rooms with what? Can anyone guess? With blood, exactly. If this was our ventricle, right? But imagine those walls are not static as they are, but are flexible, if you will. And water are coming in. If more water will come in, they will push those doors. Not the doors, the walls and the ceiling and the floor. That's exactly what's going to happen. So we're going to talk about the EDV itself, the blood that is going to stretch, if you will, the muscle wall. So increase in EDV, meaning increase in preload. Again, all of this is before we contracted. So let's see how it works, right? And you have blanks, so let's go over it and check to see everything. We'll start with the bottom here. One example would be exercise. I mentioned already last time, when we exercise, more blood is being pushed back from the tissues into the heart. We call it the venous return, the blood that comes with the veins. So more blood is coming back to the pool, to the atria. More blood will come to the ventricle. So increased venous return, there is an increased ventricular filling coming from the atria, before contraction of the atria and after contraction of the atria, right? So what about EDV? Guys, I want to hear you all. Is that going up or down? Or staying? Up or down? Up, right? Great, I just increased the EDV. By itself, before we talk about Frank Starling's law, did we already increase the stroke volume? Yes. Did we increase the cardiac output? So just by increasing the venous return, we increase the EDV. But that's not the end. Because the fact that now we have more blood in, it pushed the walls of the muscle up to a limit. And now we have an increased preload. There's more stretching. And if there is more stretching, there's an increased ventricular stretch. That's a Frank Starling's mechanism. There will be an increased force of contraction. The muscle will work harder. So not only that we start with more blood to begin with, but we do a better job in injecting blood. So what's going to happen to the ESV? Going up or down? Think about it for a second. I'm going to count to 3. ESV, 1, 2, 3. Down, exactly, because I'm pushing more than the usual. It's not only that I start with more, I could have just contracted the same, it will still be more being ejected, but no, I'm doing a better job, a stronger job in contracting. So we're going to increase EDV, decrease ESV, so it's either or, it's actually either and, right? It's both of them. And now as a result, I have an increased stroke volume, which means, of course, increased cardiac output. I mentioned last time the terms intrinsic and extrinsic. I want you guys to look at this for a second, chat with your neighbors, and when I count to 3, I want you to tell me whether this is an intrinsic mechanism or extrinsic mechanism of regulation. 1, 2, 3. This is intrinsic, exactly. It's not the nervous system, it's not the endocrine system, it's our own system because it's venous blood rhythm. The cardiovascular system itself is positively regulating itself. Any questions about preload? Yes, in the back.
So with just like normal muscles, we talked about how there was a certain point at which it wasn't beneficial anymore. So does that happen with the heart?
So great question. It won't happen here. We won't have stretch at the level that there's no overlap. There is a limit to how much you stretch the heart. Okay, that's preload. Let's talk about contractility. Contractility, the amount of force produced during a contraction at a given preload. So preloads can be different, right? But let's say there is a given preload. You stretch or you didn't stretch, or you stretch a lot even, that increased. But at a given preload, if we take 2 hearts, the same preload, if we want one of them to contract even more forcefully, can we do that? The answer is yes. And the feature is called, the phenomenon is called contractility. So before we talk about contractility, can you think of a way? I'm going to give you a second to think together. Let's say based on what we learned, especially based on what we learned about, cardiac and skeletal muscles, without going to look on the next slides, check to see if it was up to you, in your response, you were like an engineer. And the heart is a machine, so we can think about it as engineers, right? So what would you do to make the muscle contract more forcefully? Never mind how exactly you do, but what will be your goal? What will be your mechanism to do that? Not the details. You can think about a skeletal muscle. That will help you. How can a skeletal muscle act more forcefully? What more of the usual we need doing? Force produced during a contraction at a given preload. Okay? And if we talked about a contraction of the muscle at a given preload, what are we doing here? What are we changing? What will be the ESV, for example? It's going to go up or down if I increase the contraction at a given preload? Up or down? Down, wonderful, right? So that will, of course, increase the ESV. So, first of all, let's talk about systems. That gives you a clue that it's not just going to be the cardiovascular system. Which 2 systems do you think we're going to talk about? Endocrine and nervous, right? System. Okay, how would it increase contractility? That's what I just asked you. What would make the cardiocytes contract more strongly? Anyone wants to suggest ideas? I don't need the details on how you do that, the concept. What do you want to get more? Should I count to 3? Maybe you'll throw a word. Let's see if it works. 1, 2, 3. Don't be shy. I heard the word over there. 1, 2, 3. What do we need? Calcium. We need more calcium. Remember the skeletal muscle that we discussed? Myosin heads bind to actin filaments, right? The more calcium you have, the more binding sites are available, the more myosin heads will bind to actin filaments, the more forcefully you will have. So if you're able to find a way to add more calcium to the contractile muscle cells, that will probably help contraction. That's exactly the case. So we have the nervous and the endocrine system that work together on this. Okay? And go back to our endocrine and nervous system, and we learn that they can actually work together. This is all autonomic. It's the heart, the cardiovascular, the cardio muscle. Right? And we talk about the sympathetic because we want to make it stronger. You want to pump blood more. That's fight or flight. Okay? And we're going to work with the sympathetic nervous system that works directly and also indirectly with the adrenal medulla. Sounds familiar? Back in the days, before fall break, in exam 2, remember? Great. You want to forget it, but we learned about it. Okay, so sympathetic nerves secrete a neurotransmitter. Actually, it's 2 neurotransmitters, one mainly, but the another one. Which ones? I'm talking just about I'm not talking about the 2 on the chain. I'm talking about the final one. The one from the last. Which one? Norepinephrine. Great. Norepinephrine and some epinephrine, but mainly norepinephrine. What about the adrenal medulla? Which hormones does it secrete? Same, yes, same. Okay? Norepinephrine and epinephrine. So to remind you, we have the sympathetic nervous system. Part of it is going directly to the muscles, and please know, this is the red part, it's the muscle. We're not talking about the nodes here. We're not talking about rate, we're talking about contractility. As well as indirectly through the adrenal medulla, secreting acetylcholine here, but the adrenal medulla as a result will secrete hormones. A wonderful example, remember what I told you, we're using the heart as a model, basically, a paradigm to understand regulation. So this is a wonderful way to see the regulation we discussed already. One will be quick and short, one will be a bit longer, more prolonged to the blood, but reaching eventually the same targets. And what will they do? They will open calcium channels. So the result will be opening of calcium channels in the cardiac muscle, increase of intracellular calcium coming from the ECF into the cells, more calcium, more myosin-actin interaction, more pulling, stronger contraction. That's contractility, the given preload. Any questions? Intrinsic or extrinsic? Extrinsic, great. We cannot do it by itself, by the cardiovascular system itself. We need input, okay? And this is, of course, coming from our brain, basically, central nervous system. It can be something you think about. I need to fight or flight. It can be something that does not involve the cerebral cortex, okay? I am frightened by something right now. Amygdala, your hypothalamus going down, autonomic nervous system. We don't think about it. We're not aware even of that, but it's working. Today I have chickens in my backyard. Today, this morning I was just about to leave. One of the chickens, some of the chickens where I could hear them that they are really frightened. One of them was out, and there was the hugest hawk I ever saw, just standing in front, about to tell him, "You're my breakfast." And the chicken was so nervous, you could actually see the epinephrine and the calcium getting into the muscle. The chicken made it. It was able to escape. I was actually running after the hawk. I don't know what's going to happen when I come back home. But that's what happens in nature, right? That's exactly the case. There's no time to be aware or not to be aware. You need to pump more blood right now to the muscles so you can escape into the coop, and that's it. That's how it works. Okay. The third factor that regulates the cardiac output is actually in red labeled because it's going to inhibit, and that's the afterload. And that's something you guys are actually familiar with, not with the term, but with the mechanism. Afterload is the pressure that the ventricle has to overcome to force open the Cemaluna valve. I want someone here to remind me, when did we learn about this? What did we say about this? Although we didn't mention the term afterload. When did we even talk about this? It was part of the cardiovascular system discussions. At the end of this time, I'm going to need to finish. Yes? Right? Perfect. Okay, remember me being in the ventricle. We are now in ventricular systole, early ventricular systole. We are putting pressure, but the door, the semiluna valves are not opening yet. And we ask why, because the pressure outside was greater. Now if I move now to the aorta, and I'm the pressure leaning against the door, we ask where is that coming from? And we say it's coming from the previous cycle, right? That pressure, the pressure that leans, if you will, on the door from the other side, that basically makes the ventricle work harder, right? So that's what we call the afterload. The greater the pressure, the more difficult it is to pump blood at a given time. Now, the pressure here, and this is a zoom in of the semiluna valves, the aorta is right here. We call this the arterial pressure. Don't be confused with atria, arterial from the arteries. And we will start discussing this today when we'll discuss the vascular system. These are the semiluna valves. There's a pressure here, and the ventricular pressure is opposing that. We can open the semiluna valves only when the ventricular pressure will overcome, will be larger, will be greater than the arterial pressure. So that's the afterload. Where do we get this pressure? This is what we call our arterial pressure or blood pressure, as we will discuss later today. So afterload is the pressure that the ventricle has to overcome in order to force open the semiluna valves. It results from the blood pressure in the aorta. In other words, how strong the backflow of the blood came, the aorta pushed the blood towards the tissues, but some of it backflowed back into the semiluna valves, pushes the valves. Let's try to do what we did earlier on with the scheme, but this time you guys are going to do it by yourself. Okay, so I'm going to give you a couple of minutes. Try to complete everything we see here. We're starting with an increased arterial pressure, again pressure inside the arteries. Let's see the impact on the afterload, same increase, decrease, and then just continue all the way to ESV, EDV, and so on. Do it together. I'm counting to 3. First of all, you need to say either increased, decreased, or the same about the afterload. 1, 2, 3. Perfect, right? Increased afterload. So let's just follow. Let's see if we got it all. There's an increased afterload, which means that the muscle works hard. The muscle is still contracting the same to eject blood. But it's harder, there's a greater resistance. So at any point, at a given heartbeat, you will push less blood outside. So that means the ESV, remember ESV is not what's leaving, it's what's left in the ventricle, is going to increase, right? You're going to stay with more than the usual. As a result, there's going to be a decreased stroke volume. At a given heartbeat, you ejected less. What about cardiac output? Going to go up or down? Up or down? Down, exactly. So ESV is going to go up, SV is going to go down, and cardiac output, you can just add it, is going to go down. Any questions about preload, Abby?
So if the ventricle has to contract more to generate enough force to like go through the valves, wouldn't it have an increased ESV because contracting?
So you're already talking about how the muscle will respond to an afterload, if it's going to contract more forcefully. Now to any change, and by the way, in any of the factors, we might see some response of the heart to a given situation, compensation, if you will, any sort of response, right? But the heart, that will happen only after the heart muscle responds. Maybe in the next cycle, maybe in a few cycles from now, when maybe there is not enough blood going to the tissues, and someone, we will talk about that, someone will tell the brain, hey, the blood pressure here, we have a problem. We increased it to begin with, but now we don't have enough pressure. So we will have to, any question, and I will ask you a question like this, what will happen if we increase this or decrease that? Don't think about what will happen in response after a couple of heartbeats or a couple of cardiac cycles. Think about the immediate impact on the current cardiac cycle. Okay? So the current cardiac cycle, the SV will go up because we're not going to push or eject as much blood as we usually do. Any other question? And you will always have an afterload because there's always a backflow. It can go up or down, but afterload is a given. Intrinsic or extrinsic? Intrinsic, it's the blood system, right? If you cut your head off and you cut, don't do that, but endocrine system, nervous system, you still have the same regulation. Okay, so these are all the three. Go back to them later on, do that. There's a lot of stuff going on here. The only reason I'm showing it is not to confuse you actually, but for some students it's just confusing. For others it actually can help. So when you study this material after the class, when you think about it, one way to think about it is to put these schemes, right, and to see plus indicates promote or stimulate or increasing, minus the opposite, decrease. So just example of something we did. The blood volume, if we increase venous return, right, that increases end-diastolic volume. By itself, that is a plus, increasing stroke volume, which increases cardiac output. But we also learned that EDV has an indirect effect because it's increasing the preload, which decreases the end-systolic volume, which decreases the stroke volume. Minus times minus is what? It's a plus, right? So we can think about preload as promoting or increasing the stroke volume, which increases the cardiac output. If this helps you to think this way, good for you. If it's confusing, forget about this. Just find your own way. But ask yourself all the time, what if questions. What happens if one is increasing and the other one is increasing? And sometimes you cannot really tell something like an exact answer because it depends on how much. Maybe the afterload came up, but there's another impact and so on and so forth. Okay, heart rate. Let's talk about heart rate. We just talk about stroke volume. How do we increase, if you want to increase the heart rate? First of all, which systems will do that? Will do that? Nervous system, which nervous system system? Which nervous system system? Which nervous system division?
Sympathetic.
Sympathetic, right? We're talking about the sympathetic, the autonomic, but sympathetic, right? Now, what would be your target if you want to increase the heart rate, not the muscle contraction? Who is determining the heart rate? Yes, I know, exactly. This is your target. So, we're going, if sympathetic, we're going to the S as well as the AV, but it's mainly going to be the SA. Okay, that's the green here. So we saw that the sympathetic is from the middle of the spinal cord. It goes A for the muscles, that's what we already discussed about contractility. But now we're focusing on this branch that goes to the SA node and the AV node, mainly focus on the SA node. That's the green one, that's the sympathetic. So sympathetic innervation releases norepinephrine, depolarizes membranes of the SA nodal cells, raises heart rate. So you just go and do your EPSP. We don't call it EPSP because it's not another neuron, we just call it depolarization. Basically, you're changing you're depolarizing those pacemaker cells, and they reach the threshold faster than usual. While the parasympathetic will work antagonistically and will release acetylcholine right here from the bottom of this is the cardiovascular center from the medulla oblongata, going to the same target, release acetylcholine, hyperpolarizes membrane of the SA nodal cells, and lowers the heart rate because it makes it harder for the cells to reach the threshold. And it's a game, tug of war between those two. They always work when needed. When we talk about a paradigm, this is a wonderful example of the autonomic nervous system. Remember, the somatic nervous system, we tend to switch on, we tend to switch off. Lights on, lights off. This is not what works here. You don't need the nervous system for the lights on, lights off. They're always on. It's all about pacemaker cells. But it's like that fine-tuning, a little bit up and then a little bit down. Oh, this is too much, too big of heart rate. I'm running outside, I need to increase my pulse, I need to increase my heart rate, but I don't want to do it all the time, so I want to decrease it immediately after that. That's ongoing 24/7, 365 years, and for you whole 120 years. Any questions about how we regulate heart rate or stroke volume? And of course, I almost forgot, we need to mention, just imagine yourself from the green, another line here, the adrenal medulla that works with the sympathetic nervous system. We talked about it, the same concept. Okay, that's kind of like the summary of it all. Any questions about how we regulate the different cardiac output, stroke volume, etc., before we move and start to talk about the vascular system? Okay. Yeah, Ash. The reason I highlighted Ash is asking about why it's red is just to register that contractility as well as preload are increasing. Think about the blue as promoting, red is inhibiting, just to help. Okay. So this is just summary, and we will talk about the blood vessels now. Okay. It's been a while. It took us scientists, many years after we learned about the heart and the fact that there is blood to really understand the idea of circulation. Back in the days, okay, this followed all the way to the mid or so 17th century, people knew that there was a heart. They imagined that the heart is kind of like a vitality center. Okay? You eat, the food eventually is converted to life, to blood. Okay? It's going from the heart to the tissues and being consumed. The vitality is being consumed by the tissues that use that. So we just utilize the blood. We need another dish, another bunch, another batch, right, of blood. Where do we get it from? What do you think? We eat more, right? Nobody knew anything about volumes and such. It was all about observations. And only in the middle 17th century, scientists started to say, we should do some experiments, not just observation. It all came from Galen, the Roman scientist or doctor, that basically led the dogmas, and they did not want later on to do any sort of experiment. They just wanted to follow his dogmas. And they were happy with that until the, again, 17th century, until we started to see some scientists who decided, let's do some experiment. And they did mainly with snakes, but other animals and other model organisms. And what they started to do is to tie some parts of the system and to see how much blood is going into the heart, out of the heart, and so on. And then they learned about the stroke volume and cardiac output, and they realized, okay, this is 5 liters per minute in a human being. We don't eat as much, right? We cannot supply 5 liters of new blood every minute. This is a lot of eating, right? So they realized there's some sort of circulation. The system is working by itself. There's a given amount of volume of blood. Still at that point, they were not able to see the circulation itself because they didn't have a microscope at that time that were able to see the small capillaries. Okay? But eventually they were able to do that, and now we learn about circulation. So let's talk about circulation. First of all, just a true false. Learning catalytics. So I mean, isn't the arteries the ones which produce oxygen? Andrew. We need to remember that we have actually 2 circulatory systems. It is false. Okay, thank you. Common misconceptions. Okay, if you ask like 10 people in the street, none of them will probably give you the true answer, and it's false. Now, you did not do your readings yet, and everything will be for Thursday, but we will already start to talk about the vascular system, and we need to remember that we actually have 2 circulatory systems. And typically when we talk about circulation, including in this class, we will focus mainly on the tissues, what we call the systemic circulation, from heart to tissues and back to the heart. But hey, we have heart going to the pulmonary, going to the lungs, right? So how do we define arteries? Anyone know? Away from the heart, exactly. So arteries carry blood away from the heart. Veins carry blood towards the heart. Whether they carry oxygen, rich oxygen or rich in CO2, that depends on which circulation. This is a circulation, so we need to find something that connects them. And the something is what we call capillaries, or we call them capillary beds, because typically capillaries do not go as singles, they go as networks. Okay, and they are the capillary beds. Capillaries, of course, also have another very important role, which is they are responsible for diffusion of nutrients into the ECF and outside of the ECF, so our tissues can get, or the lungs, depending on which case, can get the nutrients and get rid of waste. And this is done through the capillaries. Capillaries do not reach the actual cells, they reach the ECF, or interstitial fluid. We use that term a lot, the same thing, interstitial fluid. Stop me with any questions. Again, I know we didn't read yet about it, or most of you did not for Thursday. We will just start. So the learning objectives will be these: to compare and contrast the structures and functions of the different types of blood vessels. This is what you guys are going to be responsible for in your readings, basically. We're not going to go into all in each and every step and tiny layer, but more of the concepts. We'll talk about the main groups of blood vessels, but this will help you learn better the physiology. Here, we will talk about determinants that influence the blood flow in the circulatory system. We will talk physiology, but of course, as you know, function follows form, we'll see differences. Blood, to teach and to learn, to study the vascular system, the physiology of the vascular system is a challenge, okay? It's one of those challenges where, because we don't teach or learn all the way to the most complex and complicated part of the system, sometimes it's actually harder to know only part of the picture. This is, to understand the vascular system, you really need to go into deep physics to understand fluid dynamics and so on. We're not going to do that. As always, we're going to do much more than just scratching the surface, okay? But what I'm trying to tell you is that there is a chance that today, at the end of today's class, or maybe at the end of Thursday, you'll complete the class and you'll say, I didn't get it, or, oh, I think I get it, now I'm totally confused, okay? Don't worry so much about it. Just go back, study, and process, okay? So the way I'm going to teach the cardiovascular system, the vascular system, is we're not going to think about it necessarily as a blood system. We're going to think about it following the machine in the cardiac. We're going to think about it as a water or sewer system because it's basically almost the same. So this is a different diagram than our body because this is not the cardiovascular system actually looks like, but it will be helping me to teach you. So in this case, we are looking at the heart, but we split the heart into two parts. The left heart that goes to the sends blood to the tissues, and the right heart, right, that goes to the pulmonary circulation. We have the left heart, of course, the left atrium, and eventually the left ventricle, the heart, the muscle. And then you have the aorta and the large arteries. Arteries, as we call them, elastic arteries. They can expand, but they can also be elastic. They go back to the resting status. They don't collapse. They recoil. And we'll talk about this even today, maybe. These branch into smaller and smaller arteries until they branch into small, what we call arterioles. You'll read about more arterioles later on in your readings. And those arterioles, represented here by 4 arterioles, you can actually determine their diameter. Can anyone guess? Again, you didn't read about it, so maybe you know, or even guess. How can we determine the diameter or the radius of those arterioles? Maybe smooth muscle. That's exactly the case. They're enwrapped with smooth muscle. That's part of, not enwrapped, but this is part of the arterioles. They are very rich in smooth muscles. We can find smooth muscles in others, but they are, relative to their size, they are very rich in smooth muscles. So when we're going to contract those two smooth muscles, we're going to do what? Dilate, expand, or narrow, constrict.